we start to study atom, and molecules can be formed by way of a chemical bond between atoms. And these molecules can link with other molecules or same kinds of molecule to form macromolecule. Macro means big, great, giant. So today you will study macromolecule. There are 4 types of macromolecule. One is carbohydrate, lipid, protein, and nucleic acid. Today you will mainly focus on carbohydrate. Before we start to study 4 kinds of macromolecule, I will briefly introduce some chemical concept named organic molecule. Organic versus inorganic molecule. Organic molecule always contain carbon. Often, many carbon atoms are connected in a backbone or skeleton. Organic molecules contain also oxygen and hydrogen. Many organic molecules are synthesized by organisms. And also many organic molecules are quite complex. It forms chain structure or ring structure or some helical structure. Compared to organic molecule, inorganic molecule does not contain carbon atom. So they lack a carbon backbone, and many inorganic molecules are simpler than organic molecules. Then why carbon's structure makes it so important in biological molecule? Let's think about that. So look at this carbon structure, and then a carbon atom can form up to 4 chemical bonds because it has the second shell. They only has 4 electrons, and then they want to have 4 more electrons so that the second electron shell can be fulfilled. Therefore, carbon atom can be very reactive, and then based on this structure, carbon want to participate in chemical reaction highly actively. So this property allows the formation of a complex structure such as chains, branches, and rings. And this is example, carbon can form with a chemical bond with other atoms. So you can see these 4 electrons in outermost shell that can accommodate 8 electrons via covalent bond. This provides for great diversity of molecular shape and biological molecules. So sometimes this methane can have 4 covalent bonds, and in case of carbon dioxide, even here is 4 and then here is 4, so total 8 electrons can fulfill the second electron shell. And in case of hydrogen cyanide, one side of carbon forms 6 electrons shared together, and then the other side of carbon has 2 electrons shared with hydrogen. Even though each carbon has shared electron with the other atom, but the pattern is different. Therefore, you can feel that how much this carbon molecule, carbon atom can show great diversity of molecular shape and biological molecules. Functional group, you study atoms, but some atoms is really reactive, and these atoms group together and then they highly participate in chemical reaction. We call them functional group. Functional group commonly occurring atoms or groups of atoms. Functional group attached to carbon backbone of organic molecule. Functional groups also less stable than carbon backbone, so more likely react and then more actively participate in chemical reaction. And this table shows that several kinds of a functional group. So here's name of functional group: hydroxyl group, carbonyl group, carboxyl group, amino group, and methyl group, phosphate group. When you look at the structure of each group, then the hydroxyl group is oxygen and hydrogen form chemical bond. And then you can see this hydroxyl ion when water molecule is dissociated to hydrogen ion and hydroxyl ion. This hydroxyl group is very active. Its characteristic is polar and then actively participate in chemical reaction. I do not mean you need to memorize all of these functional group, but this hydroxyl group and carboxyl group and amino group, they are important. These will come back later. In case of a carboxyl group, it has carbon double bond with oxygen. Also, carbon has chemical bond with hydroxyl group. Actually, here's COOH that indicate carboxyl group, but this is ionized form. This carboxyl group contribute hydrogen to the solution. Therefore, this table shows ionized form of carboxyl group. Another functional group, amino group, you can see nitrogen. Nitrogen has a chemical bond with two hydrogens. We call that amino group. So let's think about this question. Carbon's ability to bond with up to 4 other atoms is significant because the answer is B. The carbon is the basis of organic molecule due to its ability to form 4 covalent bonds. This property allows the formation of a complex structure such as long chain, branches, rings, and helix. So the answer is B. You need to know the terminology of monomer and polymer. So most fundamental unit of a non-living thing was atom. This atom bind together to form molecule. This molecule is singular, simple form. Simple form of a molecule can link with identical types of molecule. And then the size is going to grow up. Many molecules bind together, then we call that polymer. Poly means several, many. Mono means one, single. So monomer means small organic subunit. So this monomer kinds of a building block. So monomer bind to another monomer fuse together, it forms polymer. So the building block of polymer is monomer. Polymers means chains of monomer. When monomers fuse together, link together, then those chains of monomers are called polymer. Polymers formed by dehydration synthesis reactions. Polymers also broken by hydrolysis reaction. So you need to know about what kind of chemical reaction of dehydration and hydrolysis. So we study 4 types of macromolecule and 3 kinds of macromolecule can be classified into monomer and polymer. Today you will study carbohydrate. Carbohydrate belong to polymer. But this carbohydrate is composed of monomer named glucose. So one glucose molecule, this is monomer. But when this glucose molecule fuse together, another glucose molecule bind together. So these chains of glucose molecule eventually make polymer. Not only carbohydrate, protein also has monomer and polymer. In other words, protein is polymer. But this protein is made up of many amino acids, and each amino acid is pertaining to monomer. Same thing, DNA, deoxyribonucleic acid, and then DNA is also polymer. But this DNA is composed of many single nucleotides. So one nucleotide, another nucleotide, they join together to form polymer named DNA. But lipid we cannot categorize into monomer and polymer. The chemical structure of a lipid does not allow to separate between monomer and polymer. But the rest three types of macromolecule, carbohydrate, protein, DNA, they all shows polymer. Each macromolecule is composed of monomer. I told you that just before there are two kinds of chemical reaction named dehydration and hydrolysis. First, what is the meaning of dehydration? Hydration means what? Water. Dehydration means, D means what? Get rid of water. Okay, so this water molecule is going to be removed. Removing water to put together. Once water molecule is removed, two molecules can join together. Monomers joined. In order to make a polymer, monomer need to join, another monomer join, and whenever two monomers join, then one monomer lose hydrogen ion, the other monomer lose hydroxyl ion. Therefore, this hydrogen ion and then hydroxide ion, they form water molecule. So monomers link covalently at open electron site and water is formed. Outcome is producing polymer. So this is just monomer. So the left side is already showing very short polymer because three monomers link together. So this is short polymer. And when this short polymer bind with another monomer, this is only single monomer, then hydrogen ion from short polymer and hydroxyl ion from unlinked monomer, they can join to form water. Then once water released, then these two chemical compounds can join together. Therefore, this is a longer polymer. Before reaction, it was a short polymer, but this short polymer add one more monomer, then the length of chain is longer. This is a dehydration reaction. So you just look at the naming, dehydration. Hydration means water. Dehydration, get rid of water. So you can see that between two molecules join, at the time water molecule come out, then these two parts of molecules can link together. We call that dehydration reaction. What is hydrolysis? Now, hydro means still water. This time, lysis. Lysis means what? Break. So hydrolysis means water break apart. Dehydration reaction require when monomer to form polymer. But polymer can be break down into monomer. At the time, hydrolysis require. So polymer broken down into its original subunit, such as monomer. At the time hydrolysis take place, water donate hydrogen ion to one subunit and hydroxyl ion to the other. Energy stored in chemical bond is released. For example, digestion, when you eat chunk of food, it should be break down throughout GI tract. In other words, the polymer need to break down into smaller subunit. Water require. In other words, water donate hydrogen ion to one subunit and hydroxyl ion to the other subunit. That's why once you eat food, you need to drink water. And this water enter the body and then participate in chemical reaction. What kind of chemical reaction? Hydrolysis. This water molecule break down polymer into monomer. At the time, energy is released. So this is polymer. Okay? And then once water molecule is added, then this polymer disassembled to monomer. We call this chemical reaction hydrolysis. Chemical formula of carbohydrate is this one, CH2O. So approximately ratio between carbon, hydrogen, and oxygen is 1 to 2 to 1. This N means how many monomer you have, then the number of N can be increased. So glucose, in case of glucose, N equals 6. Therefore, C6H12O6. This is the chemical formula of glucose. The monomer of carbohydrate are small hydrophilic sugars. So carbohydrate is polymer. What kind of monomer to form polymer carbohydrate? Monomer of carbohydrate is called monosaccharide. Saccharide means sugar. So monosaccharide means just one sugar. We call this is the monosaccharide represent monomer. Disaccharide, di means two, mono means one, di means two. Therefore, two monosaccharide joined together. We call that a disaccharide. Two monosaccharides is called disaccharide. But when these saccharide, monosaccharide bind together, then they form polysaccharide. In other words, many monosaccharides. So, structure of a monosaccharide first. The number of carbon atoms varies in monosaccharide. Some monosaccharide hold 5 carbon in their structure. Some monosaccharide hold 6 carbon. When monosaccharide has 5 carbon, it is called pentose. So pentose sugar molecule carries 5 carbon. Example is DNA and RNA. They has sugar component. The sugar component of DNA and RNA are 5 carbon, pentose. So ribose act as a building block for nucleotide. So there are 3 components required to form DNA and RNA, and 1 out of 3 components is sugar, such as pentose, 5 carbon. Except DNA and RNA, the rest of carbohydrate, the rest of monosaccharide, they have 6 carbon. We call that hexose. For example, glucose, fructose, galactose, they have 6 carbon in their chemical structure. Sugar exists in the form of a ring structure in the aqueous solution. So let's look at the structure. Hexose is 6 carbon. When monosaccharide has 6 carbon, it is called hexose. So this is a chemical structure. This is a linear form. This is a ring structure. When you look at carbon here, when the line meet to form the corner, that is the location for carbon. 1, 2, 3, 4, 5 here, and 1 more, 6. So monosaccharide has 6 carbon, which is called hexose. Here's start from 1, 2, 3, 4, 5, 6. When monosaccharide has 6 carbon, that belong to hexose. And then this is a linear form. You can count the number of carbon, then it will end up in 6 carbon. So the example of glucose and fructose, these are monosaccharide, and then they have 6 carbon. Also fructose and galactose, they also have 6 carbon. 1, 2, 3, 4, 5, 6. And galactose also 1, 2, 3, 4, 5, 6. So they are the example of hexose. Hexose monomer. What about pentose? They have how many carbon? They have 5 carbon. So if we look at the structure, 1, 2, 3, 4, 5 carbon, pentose sugar. And here, 1, 2, 3, 4, 5 carbon. This is the structure of RNA. The left panel shows the structure of DNA. So DNA and RNA has 5 carbon called pentose sugar. The rest, glucose, fructose, galactose, they have 6 carbon called hexose sugar. Now this is RNA. What is the main difference between RNA and DNA in terms of sugar structure? DNA. DNA means deoxyribose nucleic acid. RNA is a short form of ribose nucleic acid. Only difference is here, this word, deoxy. DNA has the word deoxy. But when you look at this deoxy, oxy means oxygen. D means what? No oxygen. So when you look at a second carbon position between RNA and DNA, DNA does not have oxygen here. The missing oxygen atom. That's why no oxygen. Deoxy means no oxygen at second position of carbon. That is the main difference between DNA and RNA. The rest part are same. But when you look at this RNA on a second carbon, you can see here's oxygen. So RNA just called ribose nucleic acid. But DNA, according to lack of oxygen at second carbon, DNA named deoxyribose nucleic acid. But keep in mind DNA, RNA belong to pentose sugar. This is just the chemical ring structure of hexose sugar. What is the structure of disaccharide? So far we studied monosaccharide. Disaccharide can be formed when 2 monosaccharides are linked together by dehydration synthesis. So whenever molecules join, dehydration reaction involved. So hydrolyzed and converted into glucose when energy is needed. So here's glucose and glucose joined together. We call that maltose. Maltose belong to disaccharide. Here's only 2 sugar molecules join to form maltose. So maltose is disaccharide. When glucose join to galactose, then we call that lactose. Lactose is also example of a disaccharide. The milk sugar. When you drink milk, you can take lactose. Glucose joined to fructose, then we call that sucrose. This sucrose is table sugar. So this glucose, galactose, fructose, they are all monomer, monosaccharide. But 2 monosaccharides bind together, then it forms disaccharide. And what kind of monomer bind together? That result in different kinds of disaccharide. And these disaccharide can be broken down into monosaccharide this way when the body need energy. At that time, what kind of chemical reaction involved? Hydrolysis reaction require. So let's solve this question. What molecule is removed during dehydration synthesis? Dehydration itself gives the answer. Hydration, water. Dehydration, get rid of them. So one molecule will be removed. Water molecule will be removed. Let's see. That really happened. Dehydration really happened. So glucose and glucose bind together. At the time, water molecule come out to form maltose. Maltose is disaccharide. So as you see, when two monosaccharide molecules bind at the time, water molecule remove, we call this dehydration synthesis. So this is example of disaccharide synthesis. So glucose bind together at the time water molecule come out. We call this dehydration. Then the outcome is formation of maltose. The bottom panel shows glucose this time bind to fructose. Also water molecule is removed and sucrose molecule is formed. This sucrose molecule is table sugar. So, in chemical reaction, when molecules bind together at the time, dehydration takes place. Now we study monosaccharide, disaccharide. Let's move to polysaccharide. Polysaccharide are chains of monosaccharide. Since polysaccharide is big, they do not dissolve readily. The example of a polysaccharide is starch, glycogen, cellulose, and chitin. Starch consists entirely of glucose monomer. So many glucose molecules bind together, it forms starch. So this hexose sugar indicate one glucose. Many glucose bind together and it forms starch. When glucose molecule bond can branch it up to 500,000 glucose subunit. So you can see this branch occur, here branch, and then here branch occur, and branch occur here. And main function of starch is energy storage. And starch is formed in plant root and seed. Does the human body store starch? You eat starch. When you eat potato, that means you are taking starch. But once you eat starch, does the starch stay inside your body? And then human body have capacity to store starch? Think about that. Another kind of polysaccharide is glycogen, a polymer of glucose and much more branched than starch. In human, glycogen is stored in animal liver and muscle. When you start exercising, enzymes begin breaking glycogen into glucose monomer. Then this process occurs in muscle cell and then energy can be generated. So glycogen can stay inside the human body, especially where? Liver and muscle. So human body can store glycogen, but not starch. Plant can store starch. Plant cannot store glycogen. But animal including human, can store glycogen as polysaccharide, but not starch. Even though you eat potato, you just eat and then you take some energy and then the rest part is gonna remove through the feces. So human body can store glycogen but not starch. How glycogen look like? So these are starch. The upper panel shows starch and the bottom panel shows glycogen. When you look at chemical structure, you can see glycogen shows more branch compared to starch. So starch can be found in the plant, glycogen can be found in the animal. Another example of a polysaccharide is cellulose. Cellulose, where you can find abundantly, that is plant cell wall. Cellulose is composed of a polymer of a glucose monomer. What is the function of a cellulose? It structurally supports the living organism such as the plant. Cellulose comprise plant cell wall, cotton balls, and in plants, cellulose is the major component of cell wall. And some vertebrate harbor cellulose digesting microbe. And cellulose may be the most abundant organic molecule on the earth. So here, the cellulose is a major component of wood. In plant tissue, especially each single plant cell carries cell wall, and then cell wall is composed of cellulose. And when you close up cellulose fiber in the cell wall, you can see it's look like a thread, this fiber. And then a lot of hydrogen bond cross-linking occur throughout cellulose molecule, but each molecule is monosaccharide. Cellulose microfibril in a plant cell wall. So when you look at microfibril, you can see many cellulose molecules. And then each cellulose molecules also make up of glucose monomer. Another example of a polysaccharide is chitin. Chitin is similar to cellulose, but chitin bears nitrogen. So far, when you see glucose, starch, glycogen, cellulose, those molecules mainly compose of carbon, hydrogen, and oxygen. In addition to carbon, hydrogen, oxygen, chitin carries one more atom named nitrogen. Chitin's function is a supporting structure. Chitin comprises arthropod exoskeleton, such as crabs, lobster, shrimp. Their outermost layer of their body is called exoskeleton. The component of exoskeletons are chitin. Chitin can be found in those cell walls of many fungi and in many animals also. So when you look at this chitin structure, you can see this nitrogen group. Okay? And then exoskeleton. Some insect or lobster, crabs, they have a very steep external surface. We call that exoskeleton. Those exoskeleton is composed of chitin. The chitin is one example of a polysaccharide. Also, chitin is used to make a strong and flexible surgical thread that decomposes after the wound or incision heals. So you can see this monomer, carbohydrate monomer, and then also each monomer bind with nitrogen group. So what could be the answer? Yes, it contains nitrogen functional group. So answer is B. As you see, bread and even some vegetables, you can easily find the component of a carbohydrate from dietary item. What is the function of a carbohydrate? So based on structural molecule, the chitin, cellulose, Peptide glycan form fibers that give cells and organisms strength and elasticity. So, carbohydrates provide structural support. Besides being stiff and strong, the structural carbohydrates are durable. This durability is related to the fact that when you eat plant cells, they pass through your gut undigested. Human body does not have enzyme to break down plant stock. When you eat vegetable, actually you are taking cellulose fiber. This cellulose fiber is hard to break down in human gut. But why these days suggest to eat vegetable together, especially when you eat the meat? Cellulose adds bulk that helps fecal material move through the intestinal tract more quickly, preventing colon cancer, constipation, and other problems. In other words, cellulose cannot be break down into monomer in human body, but these cellulose, when they pass through the gut, they usually carry some small debris attached in a wall of large intestine. So some debris can be get rid of by help of cellulose. Otherwise, just small debris, attached, stick to inner wall of large intestine, they can pile up more and more and cause colon cancer. Then those colon cancer can be prevented from taking cellulose, because when cellulose get out of your body, at the time cellulose also, okay, the rest of waste, let's go out together. And then this cellulose remove some waste attached in a wall of a large intestine. Another function of carbohydrate is cell identity. Glycoprotein, glyco means carbohydrate. Carbohydrate can bind to protein, another types of macromolecule. Glycoprotein is a protein that covalently bonded to a carbohydrate. Glycoprotein are key molecules in what biologists call cell-to-cell recognition or cell-to-cell signaling. Your body is composed of a gazillion number of cells, but each cell communicates the neighboring cell. Most cells can communicate with lymph cells. So between cells, they talk to each other. At the time, they need some sign or signal. Those sign or signal can be detected by glycoprotein. So immune system cells use these glycoprotein to distinguish your body's own cell from foreign invader, such as bacteria. So suppose your cell, one of your cell, the bacteria invade one of your cell, then your cell put some tag or flag outside the cell membrane. Then that kind of a flag or tag is composed of glycoprotein, and then your immune cell look at that glycoprotein and then figure out, what kind of carbohydrate and protein came out of this cell membrane. Then the immune system recognize, that cell is attacked. Or immune cell said, okay, that is original our body cell, and then that cell always have normal glycoprotein attached on the cell membrane, so there's no problem, and then immune cell just pass by without attacking that cell. But when the cell invaded by foreign cell, then your cell put some specialized form of glycoprotein on cell membrane. Then those glycoprotein can be detected by immune system cells. Then immune system cell attack that cell infected by bacteria. This is the way how cell to cell communicate. Each distinctive type of cell in a multicellular organism, the nerve cells and muscle cells in your body display a different set of glycoprotein on its surface. And the immune cell read those different types of glycoprotein, and then they already have information. Oh, certain types of glycoprotein attached on cell surface, that is extremely normal. It's a healthy condition. But the some cell shows abnormal type of glycoprotein, then immune cell access and then do something. As a result, each cell type and each species can display a unique identity. Carbohydrate has a function to be a component of cell identity marker. So if you look at this cell membrane, here's membrane, and so you can imagine that here's a cell, and this part is protein, and then this protein attached to carbohydrate. We call that entire structure glycoprotein. So glycoprotein form surface coating, acting as a cell's identity tag. Glycoprotein enable the body to identify self from foreign invader. So if the cell is a healthy condition, then this glycoprotein can be read as normal to immune cell. But when this cell is attacked by bacteria, then the cell put different types of tag protein. At the time, the structure or pattern of carbohydrate is different. Then the immune cell read that tag, strange tag, then the immune cell access and then figure out and then attack this infected cell. That's why how carbohydrate participate in cell-to-cell communication and cell identity marker. Again, here's same thing. Here's glycoprotein and here's carbohydrate. So glycoprotein acting as a cell identity marker, distinguishing the body's own cells from foreign cells. The last function of a carbohydrate is energy storage. Carbohydrates store chemical energy in cells. Plants produce carbohydrate by way of photosynthesis. The essence of photosynthesis is that energy in sunlight is transformed into chemical energy, and then this chemical energy is stored in carbohydrate in the form of glucose or starch. These carbohydrates are then broken down to provide cell with usable chemical energy in the form of ATP. So this is all for carbohydrate, and then you will study the rest of 3 macromolecules such as protein, lipid, and nucleic acid. And if I include the rest of 3 macromolecules, this might be too much to students because you will have an exam tomorrow. So I'm going to stop here, but the carbohydrate will be included for the first exam. So especially you pay attention to the structure and as well as the function. So structurally how carbohydrate can be classified: monosaccharide, disaccharide, and polysaccharide, and what are the example of monosaccharide, disaccharide, and polysaccharide, and then what kind of a polysaccharide can be found in plants, and what kind of polysaccharide can be found in the animal, and also what are the function of a carbohydrate. Please read the text and then practice and then study by yourself, and then you will be in good shape on the exam. So I'm going to stop here. Then in the next video, you will start to study protein, lipid, and nucleic acid. In the continuation of studying macromolecule, the second type of macromolecule is lipid. Lipids do not form polymer. Do you remember monomer and polymer? Monomer is the building block to make up polymer. The chemical structure of a lipid does not allow lipid to form polymer. So lipids do not form by linking monomers into polymer. Lipids is waterproofing, in other words, no affinity for water, so hydrophobic. Lipids are hydrophobic because they consist mostly of hydrocarbon, which form non-polar covalent bond. You will see the structure of hydrocarbon in the next slide. Primary component of a cellular membrane, that is lipid. And also, lipid forms hormone. Lipids fall into three major groups. One is fat. Second, phospholipid. Third, a steroid. We will study each group in detail. In case of fat, oil or wax contain only carbon, hydrogen, and oxygen. Fat contain one or more fatty acid chains. Long chains of carbon and hydrogen with a carboxylic acid functional group at one end. So let's look at the chemical structure of fat or oil or wax. Look at the structure of triglyceride. Can you see the glycerol backbone? And you can think this line, vertical line, as glycerol backbone. And glycerol has 3 carbon. Each carbon of glycerol link with fatty acid. So this is glycerol molecule. You can see 3 carbon, and each carbon has fatty acid connection. Triglyceride has 3 fatty acid chain. This fatty acid is composed of many carbon and hydrogen. Therefore, we call that fatty acid chain. It is pretty long. The length of fatty acid chain is different between the types of fat. Glycerol has 3 carbon, and each carbon link with fatty acid chain. So, fat, in other words, triglyceride has 3 fatty acid chain connected to glycerol backbone. This is the left part is glycerol backbone. And then each carbon, this glycerol backbone, each carbon, 1, 2, 3 carbons of glycerol link with fatty acid chain. When you look at fatty acid chain, you can see here's carbon and hydrogen. We call that hydrocarbon chain. And then you can see 3 fatty acid chain. Here's again, this diagram shows here's a purple color indicate glycerol, and each carbon of glycerol link with fatty acid chain. Glycerol is a separate molecule from fatty acid, but they can bind each other. At the time, water molecule come out. What is the name of that chemical reaction? Dehydration reaction. So fatty acid can bind to glycerol to form triglyceride. Tri means 3 fatty acid chain. The bottom chemical structure shows triglyceride, in other words, fat. And at the time, 3 molecules of water come out, which is called dehydration reaction. Here, each fatty acid bind to glycerol, then water molecule come out. This dehydration reaction occurs to the next carbon molecule of glycerol, and then the third carbon of glycerol bind to fatty acid. Fat and oil, how they are different? Fat and oil, they are similar in terms of energy-rich molecule. More than twice as many calories per unit as carbohydrate and protein. So carbohydrate, protein, they also energy source. But when we think about which molecule produce more energy, Lipids produce more energy than carbohydrate and protein. Then what is the difference between fat and oil? Fat can be produced primarily by animals. The state of fat is solid at room temperature. Carbon atoms of fatty acid chains are linked by single bond. There are two types of fat. One is saturated fat, the other one is unsaturated fat. Saturated fat is it has many hydrogen atoms as possible. So carbon atom fully bind with other molecule. It is straight and closely packed. Therefore, since it is closely packed, it forms a solid state at room temperature. Compared to fat, oil produced primarily by plants. The state of oil is liquid at room temperature. That is the main difference between fat and oil. Some carbon atoms of fatty acid chain are linked by double bond. In case of fat, all saturated by single bond. Single bond means every carbon in fatty acid has single chemical bond with the other atom. In case of oil, unsaturated fat means some carbon atoms of fatty acid chain are linked by double chemical bond. We call that unsaturated. So carbon is not fully saturated in 4 chemical bond. Fewer than maximum hydrogen bond possible, kink, and more loosely packed. At room temperature, butter is a solid state. You can see this dark gray color indicate carbon, light gray color indicate hydrogen. But when you look at oil, it is liquid state at room temperature. See, some carbon has double bond. That provide the room of fatty acid. Therefore, the fatty acid chain can flexible and they can kink. Fatty acid vary in length, the number of carbon, and in the number and location of double bonds. Saturated fatty acid have the maximum number of hydrogen atoms possible and no double bond. Unsaturated fatty acid have one or more double bond. So look at saturated fat. Can you see this hydrocarbon chain or fatty acid chain? Each carbon has 4 chemical bonds. Each carbon are fully saturated with other atom, such as hydrogen. In case of unsaturated fat, most carbon saturated with hydrogen, but some carbon is not fully saturated with hydrogen. Rather, it has a double bond with adjacent carbon. In this case, there is some space or room can be formed that allow unsaturated fat become flexible. This is the chemical structure of unsaturated fat. You can see this double bond area. The fat can have some small space to bend. Fats made from saturated fatty acid are called saturated fat, and they are solid at room temperature. Most animal fats are, this is a little bit typo. This is most animal cells are saturated fat. Fats made from unsaturated fatty acid are called unsaturated fat or oils and are liquid at room temperature. Plant fat and fish fat, they are usually unsaturated. You might hear about trans fat and omega-3 fatty acid. Hydrogenation means artificially break some double bond and add hydrogen. You see in unsaturated fat, you see this kind of double bond between 2 carbon atoms. So this has like this. So each carbon has 4 chemical bonds. In this case, 1, 2, 3, 4. Then this double bond can be broken and add. So this is gonna single band between 2 carbon atom and add hydrogen artificially. This convert liquid oil to solid. We call that trans fat. It extends shelf life but encourages arterial cholesterol deposition. Trans fat make us feel crispy. So when company synthesize trans fat, that product can maintain constant state for a long time. Once we eat trans fat, it can cumulative in artery, then it causes some cardiovascular problem. Omega-3 fatty acid. Certain unsaturated fatty acid cannot be synthesized in our body. Those nutrients must be supplied in the diet. And then fish, salmon, they really carry good omega-3 fatty acid. Omega-3 fatty acid, they are not synthesized in human body, but it requires for normal growth and provide protection against cardiovascular disease. Wax, chemically similar to fat. Most animals lack enzymes to digest wax, so we do not eat wax. Wax is highly saturated, so it is a solid at outdoor temperature. Water repellent, in other words, hydrophobic. Wax can be found on the leaves and stems of plants or bird feather or honeycomb. So wax has a hydrophobic property. So one more time you can compare. Fat can be subcategorized into saturated fat and unsaturated fat. Saturated fat is solid state at room temperature because the fatty acid chemical structure shows each carbon is fully saturated with hydrogen. So when you look at this space-filling model of stearic acid, you can see each carbon is fully saturated with hydrogen. Unsaturated fat is liquid state at room temperature. And when you look at the chemical structure of a fatty acid, there is some carbon forms a double chemical bond. When you look at this space-filling model of oleic acid, this carbon forming double bond with adjacent carbon has more room or space to bend, and this can kink. It gives flexibility to unsaturated fat. Lipid can be subcategorized into 3. One is fat, second is phospholipid. Phospholipid comprise plasma membrane. Chemically similar to oil or fat, but it has a different end of chemical structure. 2 hydrophobic fatty acids, we call that tail, and hydrophilic phosphate group are attached to glycerol. We call that head. So we can simply draw down this way. This is head part, hydrophilic region, and this is the tail part, hydrophobic region. These 2 fatty acid tails are hydrophobic, and this head region is a hydrophilic, which has phosphate group. When phospholipids are added to water, they self-assemble into a bilayer with the hydrophobic tails pointing toward the interior. The structure of a phospholipid results in a bilayer arrangement found in cell membrane. Here's head region is hydrophilic and tail region is hydrophobic. Hydrophilic, hydrophobic. Head region hydrophilic, tail region hydrophobic. Hydrophobic means dislike water. So like oil. Oil dislike water. Oil like oil. Therefore, when they form two layers, this tail and tail want to face to each other. Therefore, here's two tail and here's head, hydrophilic. Hydrophobic face to each other and hydrophilic is far. This forms bilayer, two layer. This is a basic pattern to form plasma membrane. First, phospholipid chemical structure look like this. This is glycerol backbone. There is 3 carbon on glycerol molecule. And in case of fat, each carbon of glycerol molecule bind to fatty acid. But in case of phospholipid, this is phospholipid, only 2 fatty acid chains will bind to 2 carbons of glycerol molecule. The rest carbon of glycerol molecule will bind with phosphate group. Therefore, we call that phospholipid. This phosphate group binding region has polar property, hydrophilic property. This fatty acid region shows hydrophobic property. Therefore, we can just symbolize head region, hydrophilic, and tail region. Because of 2 fatty acid tails, we can draw 2 lines, and this is tail region, and this is head region. Head region shows hydrophilic property, tail region shows hydrophobic property. One more time, this is glycerol molecule, glycerol backbone. 2 carbons of glycerol molecule bind with fatty acid chain, and the rest carbon of glycerol molecule bind with phosphate. Therefore, we call that phospholipid. This is space filling model. Here's 2 tails indicate fatty acid, and above part head region is glycerol, and phosphate portion shows hydrophilic property. So we can simply draw phospholipid this way, head and tail. If fatty acid of phospholipid has a double bond between 2 carbon atoms, then you can draw a little bit kink. If fatty acid chain do not have any double bond between 2 carbon atoms, then you can just draw straight line. But the above head region is composed of phosphate and glycerol. Head region, polar region, therefore hydrophilic. The tail region, non-polar region, therefore hydrophobic. And this single phospholipid molecule is gonna array to form membrane. The polar hydrophilic region like water, and hydrophobic tail region dislike water. So one layer of plasma membrane and the other layer of plasma membrane appears this way. Bilayer of a plasma membrane does not have this configuration. Why? Because this tail region is hydrophobic and head region is hydrophilic. They are not very friendly. Therefore, they do not like this composition. They are comfortable by forming tail face to tail, and head is far from tail region. That's why plasma membrane have these kinds of composition. Also, when fatty acid chain has double bond between 2 carbon atoms, they can have some space. In this space, some other molecules can be integrated. Some other protein can be integrated. So this is a plasma membrane. And maybe you can imagine this. This is the cell. So this is inside the cell. This is outside the cell. Inside the cell, also liquid state. Outside the cell, there's liquid also. In extracellular matrix, always have liquid state. So the head region of phospholipid, this is a single phospholipid molecule. This head region, polar property, therefore they like water, hydrophilic. That's why head region of phospholipid face to outside. What about this phospholipid also? This head region will face to watery environment inside the cell. But between these two tail regions, they are hydrophobic region. So they like each other. This configuration of a plasma membrane really make head and tail region safe and stable. The third type of lipid is a steroid. Steroids are lipids characterized by a carbon skeleton consisting of 4 fused ring structure. 4 rings of carbon atoms sharing one or more sides with various functional groups attached. The example is cholesterol. Cholesterol is a vital component of nerve cell insulation. There's an axon in nerve cell. That axon can be wrapped around by cholesterol, which we call the myelin sheath. And then that is very important in terms of conducting nerve signal. Also, cholesterol used to synthesize other steroids such as estrogen and testosterone. The precursor of estrogen and testosterone is cholesterol. So from cholesterol, there's some chemical modification take place, resulting in estrogen and testosterone. So we need cholesterol, to have estrogen, testosterone, and also, to form myelin sheaths around the nerve cell. But when we have too much cholesterol, this cholesterol can be accumulated in the wall of a blood vessel, which can cause cardiovascular problem. So although cholesterol is essential in animals, high level in the blood may contribute to cardiovascular disease. So this is chemical structure of steroid. So cholesterol, you can see 4 ring structure. From this cholesterol, there's some chemical modification take place, and then estrogen and testosterone can be formed. Estrogen and testosterone also have 4 ring structure because they originated from cholesterol. So steroid, so these are all hormone. And cholesterol, can you see this is phospholipid, head region, tail region, and head region, tail region, and these phospholipids carries unsaturated fat. Therefore, they have a little space. In that space, cholesterol can be embedded throughout plasma membrane. Depending on what kind of a hormone can be formed, determine the gender. So, female produce estradiol, eventually turn into estrogen, and male produce testosterone, but estrogen and testosterone, they can be made from cholesterol, which is not a type of lipid. Triglyceride means 3 fatty acid chain bind to glycerol. In easy name is fat. Wax, oil, peptide, which is not the type of lipid. Peptide is protein, so this is not belong to lipid. But triglyceride, wax, and oil, they are example of a lipid. Besides the three fatty acids, a triglyceride molecule also has what other chemical group? I said tri means three fatty acid chain bind to what backbone? Glycerol backbone. So the answer is A. Let's move to protein. Proteins account for more than 50% of the dry mass of most cells. Proteins have many functions. One of them is enzyme promote specific chemical reaction. And also, it forms a structural protein. For example, keratin is the protein on your hair. Nutritional, albumin and casein. Casein is a milk protein. Also, protein role as a transporter, such as hemoglobin is a protein and hemoglobin deliver oxygen and carbon dioxide. Contractile, which means when you use muscle has muscle fiber. This muscle fiber is composed of actin and myosin. This actin and myosin, they are composed of a protein. Some hormone, when you study lipid just before, steroid hormone, but some hormone can be formed by protein. For example, insulin. Insulin, the chemical structure shows protein. Or growth hormone. Some protein role as a growth hormone. Antibody is important to kill the foreign invader. This antibody has protein structure. Toxin, also some toxin has a protein structure. This table summarizes the function of a protein, structure and movement in muscle, defense, antibody, a storage, some nutrient, a signaling, insulin signaling to control the glucose level in the blood, and catalyzing reaction. Catalyzing means this protein control the speed of a chemical reaction. At the time, we call that specific function of a protein named catalyst. One of the functions of a protein is called enzyme. Only protein can work as an enzyme. This enzyme controls the speed of a chemical reaction. At the time, we describe that reaction catalyzing reaction by enzyme or catalyzing reaction by protein. So when we study enzyme, this will come back. So this is a detailed diagram to explain enzymatic protein. So here's chemical reaction occur. These are the reactant and these are the product. But enzyme really facilitate chemical reaction take place very fast with a low activation energy. Defensive protein, this is antibody. Here's antibody. This antibody grab virus. Therefore, this antibody kill the function of a virus. A storage protein, albumin or casein are very good nutrient source. It can be found in egg or milk. Transport protein, transport substance. Here's membrane. I told you that some protein integrated throughout plasma membrane, and since plasma membrane has hydrophilic and hydrophobic property, some molecules have difficulty to pass through plasma membrane. But sometimes protein integrated throughout plasma membrane and provide some channel. Some molecule which has difficulty cross the membrane can easily enter the cell. Hormonal protein, so the insulin can control the glucose level in the blood. Receptor protein, so the nerve cells communicate each other. At a time, some signaling molecule can be transmitted from one nerve cell to adjacent nerve cell. At the time, there is some receptor on the cell surface is ready to accept this signaling molecule. These receptor molecules is composed of a protein. In the muscle, when you have a movement, there is a muscle fiber contract and relax. Those muscle fibers is composed of actin and myosin. These actin and myosins are protein. Also structural protein such as collagen. For example, keratin is the protein of hair and horns, feather. And in case of collagen, it provides a fibrous framework in animal connective tissue. So protein really shows many important functions. Let's look at the structure. Proteins are polymer of amino acid. Now we go back to monomer and polymer. Lipid cannot be arranged into monomer or polymer, but the rest three macromolecules such as carbohydrate, protein, and nucleic acid, we can arrange them in monomer and polymer. In other words, monomer such as amino acid, they link together to form polymer named protein. So one amino acid link with second amino acid link with third amino acid. So a sequence of amino acid form polymer named protein. So each amino acid has central carbon and has amino group. And carboxylic group and hydrogen and R group. This is basic structure of each amino acid. Each amino acid has amino group, carboxylic group, and R group. What is the meaning of R group? Here's a central carbon, amino group, carboxylic group, and hydrogen. And the other one is a variable group, R group. So different kinds of chemical molecules can bind to R group. Polypeptide is unbranched polymer built from the same set of 20 amino acids. I told you just that these amino acids, there are 20 kinds of R group. When I introduce R, a variable group, there are 20 kinds of variable group in amino acid. That's why polypeptide can be built from the same set of 20 amino acids. A protein is a biological functional molecule that consists of one or more polypeptide. Polypeptide is a similar meaning of a protein. Amino acid has carbon, therefore organic molecule with carboxylic and amino group. Amino acids differ in their properties due to different side chains called R group. There are 20 kinds of R groups. R groups gives each amino acid distinctive properties. Some R group of amino acids show hydrophilic property. Some other R group shows hydrophobic property. Some R group shows disulfide bond. So let's look at amino acid structure first. Here's a single amino acid structure has a central carbon, amino group, carboxylic group, and hydrogen. This is R group. R group is a variant. There are 20 kinds of different R groups. So here's central carbon, amino group, carboxylic group, and 20 kinds of different R group. This is a single amino acid. So can you see this purple box? That indicates a single amino acid, and second amino acid, and third amino acid. And first amino acid and second amino acid, they already bind each other by way of peptide bond. and second amino acid and third amino acid try to bind each other at the time water molecule removed. We call this dehydration reaction. Once dehydration reaction occurs, then synthesis of molecules take place. So before reaction, there are two amino acid link. After reaction, there are three amino acids you can see. So each amino acid form chemical bond. The name of a chemical bond is peptide bond. So here's one amino acid and here's another amino acid. Two amino acids bind each other by way of peptide bond. I told you that 20 kinds of different R group. Look at the below of this colorful box. This is a central carbon, amino group, carboxylic group. Here's another amino acid, central carbon, amino group, carboxylic group. The same pattern. But when you look at R group, the R group chemical structures are all different. According to different R group, each amino acid has its unique name. In this case, if R group has this chemical composition, this amino acid is called glutamic acid. If another R group has this chemical composition, then it is called aspartic acid. Phenylalanine, leucine, cysteine. In case of cysteine, you can see some disulfide bond. This is a sulfur. S is a sulfur. So sulfur-containing functional group. The chemist already classified 20 kinds of amino acid. The purple box indicates basic structure of every amino acid. Central carbon, amino group, carboxylic group. You can find the central carbon, amino group, and carboxylic group commonly throughout this amino acid. But R group is different. Glycine has only one hydrogen in R group, but alanine has CH3 methyl in R group. Valine has this chemical composition. These yellow boxes represent R group. The top panel shows non-polar side chain hydrophobic amino acid. According to the property of R group, these amino acids dislike water. Some amino acid out of 20 kinds of amino acid shows hydrophobic property. But some amino acid shows hydrophilic property. For example, still same way the purple box indicates the common chemical structure of amino acid. So central carbon, amino group, carboxylic group. And then when you look at green box area, those are the R group. And this R group, the chemical composition of this R group shows water-like property. Therefore, hydrophilic amino acid. The bottom panel shows electrically charged side group, hydrophilic, negatively charged hydrophilic, positively charged hydrophilic amino acid. So I do not want you to memorize all these, but you must know that the common core chemical structure of amino acid is central carbon, amino group, carboxylic group. That structure can be found throughout 20 kinds of amino acid. It's commonly. But the R groups are all different. According to the property of R group, some amino acid shows hydrophobic property, some amino acid shows hydrophilic property. In case of a hydrophilic property, some amino acid highly charged negatively, some amino acid charged positively. That's it. I already told you that when two amino acids join together by way of a peptide bond, proteins are formed by dehydration synthesis. Nitrogen in amino group of one amino acid covalently bond to carbon in carboxylic acid group of another amino acid. We call that peptide bond. Peptide bond is type of covalent bond, but it specifically occurs between two amino acids. Scientists put another name, peptide bond. When 50 amino acids link together, we call that peptide. When amino acids link together more than 50, we call that polypeptide. So polypeptide means more than 50 amino acids join together, then we call that polypeptide. This polypeptide is equal to protein. One more time, this already has two amino acids linked together by way of a peptide bond. And these two amino acids is going to join with another, one more amino acid. Then here's from carboxylic group, hydroxyl ion, and in amino group, hydrogen ion, they join to form water, and this water get rid of them. Then these two reactants can join to form final product, 3 amino acids join together. And we call this peptide bond. Peptide bond. So here's one amino acid, second amino acid, third amino acid. There's a peptide bond between two amino acids. That is primary structure. Protein structure and function. Interactions among amino acid and R groups cause twist, fold, interconnection. A sequence of amino acid is called primary structure. This primary structure is going to continue to twist, fold, and has interconnection between amino acid. That generates secondary structure. A functional protein consists of one or more polypeptides precisely twist, fold, and coiled into a unique shape. The sequence of amino acids determines a protein's three-dimensional structure. We call that tertiary structure. So here's secondary structure. A protein's structure determines its function. Very important. Protein's structure determines function. There are 4 levels of protein structure, such as primary, secondary, tertiary, and quaternary structure. First, primary structure of a protein is just a unique sequence of amino acid. So amino acid can be linked by way of a peptide bond. So you have a series of amino acid that is just the primary structure of a protein. The primary structure is like the order of letters in a long word. And this primary structure of a protein is determined by DNA. So DNA already have information to produce primary structure of a protein. Secondary structure of a protein means from primary structure of a protein, there's some hydrogen bond occur within amino acid. It consists of coils and folds in the polypeptide chain. So it forms some structural pattern such as a helix or pleated sheet. You can think about this as a single amino acid. A series of amino acids. This is just primary structure. Polypeptides are unbranched polymer built from 20 kinds of amino acid. These are all single amino acid linked together by way of here's peptide bond. So look like more than 50 amino acids. So we can call this polypeptide. So just a sequence, order of amino acid. We call that primary structure of a protein. This primary structure of a protein gonna have a hydrogen bond between amino acid. Secondary structure are maintained by hydrogen bond between atoms of the polypeptide backbone. The dot line shows hydrogen bond. Segment of the polypeptide chain coil or fold into local pattern called secondary structure. There are two types of secondary structure. One is alpha helix, like this, alpha helix. The other one is beta pleated sheet, like this, beta pleated sheet. So peptide bond occur between JSON amino acid. Hydrogen bond can occur one amino acid with a fold down of another amino acid. That brings some regular pattern of structure. We call that secondary structure. Tertiary structure of a protein means tertiary structure of a protein is determined by interactions among various side chains. Between R group, there is a chemical bond can be formed. This makes secondary structure fold or more twist to form globular structure. These interactions between R groups include hydrogen bond, ionic bond, hydrophobic interaction, and van der Waals interaction. I did not mention about van der Waals interaction. There's one more kind of chemical reaction in addition to covalent, ionic, and hydrogen bond. It's a very weak chemical interaction. But when protein fold to form tertiary structure, van der Waals interaction also participate. Strong covalent bond called disulfide bridge may reinforce the protein's structure. So this is tertiary structure. You can see this linear, the ring was secondary structure. This secondary structure fold, twist more to form tertiary structure. How they can twist and coil? Because there is a chemical reaction take place between R group. Let's see. You can think this purple string, purple line as here's a central carbon of first amino acid. Here's central carbon of a second amino acid. Here's a central carbon of a third amino acid. So there is peptide bond between first amino acid, second amino acid. Therefore, this should have nitrogen and double bond with carbon of second amino acid, and we call this peptide bond. There should be peptide bond between second amino acid and third amino acid. Now this secondary structure is already showing alpha helical structure. Secondary structure of protein is going to fold, twist, and coil more severely. Then eventually this is R group of first amino acid. This is R group of, maybe this might be fourth amino acid. As a secondary structure of a protein twist and supercoiled, the R group of amino acid 1 can closely located to R group of fourth amino acid. That allows to form chemical bond between first amino acid and fourth amino acid by forming hydrogen bond. This will generate globular structure. The secondary structure was alpha helix, but as alpha helix formed, then there's a chance to R group meet each other even though those amino acids located further down. There's a chemical bond such as hydrogen bond occur between R group, it forms more condensed, supercoiled protein structure. We call that tertiary structure. From tertiary structure, the last level of a protein structure named quaternary structure. When two or more polypeptide chains form one macromolecule. Collagen is a fibrous protein consisting of 3 polypeptides coiled like a rope. Hemoglobin is a globular protein consisting of 4 polypeptides, 2 alpha polypeptide and 2 beta polypeptide chain associate together. So look at here, this is a quaternary structure. Can you see this purple tertiary structure? And behind the purple color tertiary structure, can you see pale blue tertiary structure associate? If you look at bottom, another purple color tertiary structure, and behind that tertiary structure, you can see pale blue color tertiary structure. So tertiary polypeptide 1, 2, 3, 4. 4 identical polypeptides associate to form quaternary structure. The left linear structure shows primary structure of a protein. The primary structure is a series of amino acids by way of peptide bond. There's a peptide bond between here, peptide bond between each amino acid. From primary structure, hydrogen bond can take place between amino group of one amino acid and further separate down carboxylic group of another amino acid. So these dots indicate hydrogen bond. These hydrogen bonds form a regular pattern of shape called alpha helix or beta pleated sheet. This secondary structure is going to continue to supercoiled by way of hydrogen bond between R group that generate tertiary structure. Folding of a helix, alpha helix, result from hydrogen bond with the surrounding water molecule and disulfide bridge between cysteine amino acid. In other words, hydrogen bond occur between R group of several amino acids, as well as this secondary structure of a protein will be located in watery environment. Then the water molecule also form hydrogen bond to cause secondary structure of a protein fold and supercoiled into tertiary structure. This tertiary structure is functional. In other words, it has a specific function as an enzyme. I will come back there later. But tertiary structure, when you look at this tertiary structure, can you see this tertiary structure? And here's another tertiary structure. Behind the yellow color, you can see blue polypeptide, also 2 blue polypeptide. There are 4 identical polypeptides associated together to form quaternary structure. There are 4 levels of a protein structure. Primary structure, just a simply sequence of amino acid. And primary structure can fold, twist by way of a hydrogen bond. We call that secondary structure. So do not confuse that. Primary protein structure has peptide bond. A secondary protein structure can be formed by way of hydrogen bond. Here's primary structure. Then this primary structure can form alpha helix or beta sheet by way of hydrogen bond. And hydrogen bond can be formed between R group that produce tertiary structure, so long range folding. So hydrogen bond can be formed not nearby amino acid. It's further down amino acid between R group. So it forms this like tertiary structure. It's globular shape in 3-dimensional structure. This tertiary structure can associate with identical tertiary structure to form quaternary structure. I told you that tertiary structure is very important in terms of a function. When protein cannot form proper tertiary structure, it causes disease. One example is sickle cell disease. A slight change in primary structure can affect a protein's structure and ability to function. Sickle cell disease, an inherited blood disorder, results from a single amino acid substitution in the protein hemoglobin. So normal hemoglobin has this primary structure of a protein, and this primary structure of a protein further folding and twist to form secondary and tertiary structure and eventually to produce quaternary structure. And this is normal hemoglobin. Once hemoglobin protein formed normally, then it shows like this red blood cell. It's a ring structure. At the center is a little bit collapsed. Sickle cell hemoglobin, one amino acid number 6, glutamate GLU, mutate to VAL, valine. This causes abnormal structure of secondary and tertiary structure. Further, this abnormal secondary and tertiary structure brings this structure of red blood cell. It's not round shape. It is crescent structure. Just a single amino acid change can affect the structure of red blood cell. So it's very important to have normal amino acid sequence. Otherwise, the outcome can be serious. Sickle cell disease or sickle cell anemia can cause dizziness and also several side effects. It will come back when you study inheritance. Do you remember? This amino acid sequence can be determined by DNA sequence. So if DNA sequence is mutated, that causes the changing of the sequence of primary structure of a protein. Eventually, the protein cannot be normally produced. So DNA sequence is the top stream. If something is wrong on DNA stream, then the next following primary structure of a protein can be influenced. That affects secondary, tertiary structure, eventually the final protein structure. So 4 levels of protein structure. Primary structure is just a series of amino acid. And this a sequence of amino acid is folding and twist to form secondary structure called alpha helix or beta pleated sheet. And this secondary structure further super coiled and twist between R group, and it forms tertiary structure. And this tertiary structure associate with identical tertiary structure to form quaternary structure. And this tertiary structure is very important in terms of protein function as an enzyme. Function is determined by structure. Precise position of amino acid determine secondary and tertiary structure. Determine how regions of a protein interact with each other as well as other molecules. So how certain regions of a protein interact with each other, that determine the tertiary structure. What determines protein structure? In addition to primary structure, physical and chemical condition can affect structure. So primary structure is just a series of amino acid. A sequence of amino acid need to fold in an ordered way, in an arranged way. At the time, some environmental factor can influence the folding of a primary structure. What kind of environmental factors, such as pH, salt concentration, temperature, or other environmental factor can cause a protein to unravel? This loss of a protein's native structure is called denaturation. If secondary structure is not located in proper pH environment, the secondary structure cannot form normal tertiary structure. Also, if the environment is too high temperature, protein cannot fold correctly. We call that denaturation. I told you that the tertiary structure of a protein is very important in terms of a function. The protein cannot fold properly. The tertiary structure of a protein lose its function. We call that denaturation. Disruption of a tertiary or secondary structure is called denaturation. Primary function left intact. The sequence of amino acids are not influenced, but the folding pattern disrupt, therefore protein lose function. So this is normal protein. If this normal protein located in extreme pH or high temperature, then protein cannot maintain proper chemical bond. Then it becomes change the structure. We call that denaturation. And once protein denatured, then this protein lose function. But when we change the temperature to the original state, then denatured protein can revert back to normal protein, which is called renaturation. But some protein does not go back to normal protein, even though proper environmental factor engaged. Once some protein becomes denatured, that denatured protein cannot revert to normal protein. Some protein is easily go back to normal protein, some of them not. So it depends on the characteristic of a protein. A peptide bond forms between which two functional groups? Carboxylic group and amino group. The answer is D. Let's move to nucleic acid. The last type of a macromolecule. Nucleic acids are polymer of a nucleotide called polynucleotide. So this is the concept of monomer and polymer. Nucleotide is monomer and polynucleotide is polymer, in other words, nucleic acid. Genes are made of DNA. Nucleic acid made of monomer called nucleotide. So many nucleotides to form nucleic acid called DNA. Let's look at single monomer nucleotide, the structure of a nucleotide. Nucleotide has 5 carbon sugar. We call that pentose. Do you remember when we study carbohydrate? Some carbohydrate has 5 carbon. Some monomer of a carbohydrate has 6 carbon. If the monomer of a carbohydrate has 5 carbon, we call that pentose sugar. If the monomer of a carbohydrate has 6 carbon, we call that hexose sugar. Nucleic acid has 5 carbon sugar, we call that pentose sugar. There are 2 types of pentose sugar. One is ribose, the other one is deoxyribose. So nucleotide has 3 components. One is sugar. Second one is phosphate group. Third one is nitrogenous base. Nitrogenous base, there are 4 types of nitrogenous base, and 2 of them has single ring in their chemical structure. The rest 2 have a double ring in their chemical structure. When nitrogenous has single ring, They are called thymine. So let's look at 4 kinds of nitrogenous bases. We call that simply bases. Can you see two ring structure adenine, 2 ring structure guanine, single ring structure cytosine, single ring structure thymine. So 4 kinds of base in DNA. They are adenine, guanine, cytosine, thymine. Adenine and guanine, since they have 2 ring structure, biologists put another name of subgroup, purine. The rest 2 nitrogenous base have single ring structure, so biologists put name of subgroup, pyrimidine. 4 kinds of bases in DNA is subcategorized in 2 groups, purine and pyrimidine, and purine has adenine and guanine as base. Pyrimidine group has cytosine and thymine. All you have to memorize is there are 4 kinds of bases in DNA. What are the names? Adenine, guanine, cytosine, thymine. Adenine and guanine has 2 ring structure. Cytosine and thymine have 1 ring structure. This shows 3 components of nucleotide: sugar, pentose sugar, phosphate group, and base. The role of nucleotide: critical energy carrier, intracellular messenger. So these 2, critical energy carrier, intracellular messenger, can be done by energy molecule ATP. ATP molecule belong to nucleotide because its chemical structure carries sugar, phosphate, and base. The rest role of nucleotides comes from DNA. So nucleotide is a subunit of nucleic acid. There are 2 types of nucleic acid. One is deoxynucleic acid called DNA, ribonucleic acid, RNA. The portion of a nucleotide without the phosphate group is called nucleoside. ATP, I just told you that it's energy molecule, and then these ATP belong to nucleic acid macromolecule because it has ribose nucleotide with 3 phosphate group. ATP is formed by reaction that release energy. Once carbohydrate, glucose breakdown, then ATP can be produced. ATP store energy in bonds between phosphate group, so energy can be released when one of those phosphate bond is broken. So energy drives reaction, polymerization, so ATP participate in chemical reaction by providing energy. This is the structure of ATP. One is sugar, pentose sugar, 5-carbon sugar. Second is phosphate. Third is base. What kind of a base? Adenine. That's why adenosine triphosphate. Nucleotide. This is, look at sugar, phosphate, base. Sugar, phosphate, base. Sugar, phosphate, base. So this is one nucleotide, the second nucleotide, third nucleotide. And each nucleotide join together to form sugar phosphate backbone. sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. So this one strand can be formed by sugar phosphate backbone, and nitrogenous base protrude from sugar phosphate backbone. And the other strand also has sugar phosphate backbone. Sugar shows this 5 pentagonal structure in blue color. Phosphate shows circle of yellow color. So you can see sugar phosphate backbone of the other DNA strand. DNA strand always exists double strand. 2 strand, each strand can be formed by way of sugar phosphate backbone, and each DNA strand has protrusion of a base. We can see guanine, G means guanine, A means adenine. And the other DNA strand you can see, this time cytosine and thymine. The base protruding from each DNA strand can bind each other by way of hydrogen bond. They can form DNA structure in double strand. We call that double strand helix. ATP and DNA are different because ATP is monomer. ATP work as independently from DNA. ATP has a chemical structure which belongs to nucleic acid, but it works as energy molecule. Its chemical structure belongs to nucleic acid category of a macromolecule. So ATP is monomer and DNA is nucleic acid polymer. So the answer is, so ATP is just a single molecule, but it has ribose, sugar, and phosphate, and base. Therefore, the structure make ATP pertaining to nucleic acid macromolecule. DNA versus RNA. DNA, its real name is deoxyribonucleic acid. RNA, ribonucleic acid. I think I already mentioned this. Only difference is ribonucleic acid, they are same, but DNA has deoxy. Deoxy means, oxy means oxygen. Deoxy means no oxygen. DNA has 2 strands, twisted and linked by hydrogen bond. Double helix, genetic material of all cells. DNA sequence spells out the recipe for protein. The primary structure of a protein resulted from the information of DNA. Ribonucleic acid RNA is a single strand of a ribose nucleotide. The copy of DNA is RNA. Direct protein synthesis. RNA directly participate in protein synthesis. DNA cannot directly participate in protein synthesis. So there are some difference between DNA and RNA, and I want students to know about the difference between DNA and RNA. First, in terms of structure, here, the second carbon of sugar, when you look at only hydrogen atom, that means deoxyribonucleic acid. This is the DNA structure. Deoxy, there is no oxygen. So DNA has 5 carbon sugar. On second carbon of 5 carbon sugar does not have oxygen. That indicates DNA. For RNA, the second carbon of pentose sugar has oxygen, hydroxyl group rather than hydrogen. So here's oxygen. So we get rid of deoxy, just put ribonucleotide, ribonucleic acid. So that is main difference. based on sugar structure. Green pentagon indicates sugar, and here's small yellow circle indicate phosphate, sugar, phosphate, sugar, phosphate backbone form one DNA strand and base protrude from this sugar phosphate backbone. This is a single DNA strand, so you can see 5 carbon sugar, pentose, and phosphate group, and nitrogenous base. This indicates single nucleotide. DNA always exists in double strand. So here's DNA strand. Here's another DNA strand. So if forming double strand. When you look at this single strand, here has thymine base, and the other DNA strand has adenine. So adenine and thymine, they always bind each other. Guanine and cytosine, they always bind each other. But thymine cannot bind with guanine. Adenine cannot bind with cytosine. Why? Based on their chemical structure, thymine can form only 2 hydrogen bonds. Adenine also can form 2 hydrogen bonds. Guanine and cytosine, they can have 3 hydrogen bonds. Therefore, thymine cannot have hydrogen bond with guanine because the number of hydrogen bonds does not make they fit each other. So this is the rule. In other words, when you know the base sequence of one DNA strand, you can automatically pull out base sequence of the other DNA strand. Why? Because thymine always bind with adenine, and cytosine always bind with guanine according to the number of hydrogen bonds. Which part of DNA nucleotide below is different from analogous nucleotide in RNA? Phosphate, same. Base, a little bit different, but right now we say that same. Bonding, hydrogen bond, same. But what is the main difference? Sugar. Nucleotide polymer are linked together to build a nucleotide or nucleic acid. JSON nucleotides are joined by covalent bond that form between hydroxyl group on the 3' end carbon of one nucleotide and phosphate on the 5' carbon on the next nucleotide. You can easily understand this structure when you see the diagram. These links create a backbone of sugar phosphate unit with nitrogenous bases as appendages. The sequence of bases along a DNA or a messenger RNA polymer is unique for each gene. M means messenger RNA. RNA molecules usually exist as a single polypeptide chain. RNA molecule is not stable as much as a DNA molecule. In the DNA double helix, the two backbone runs in opposite orientation each other. The arrangement is called anti-parallel. We also see this anti-parallel structure looking at diagram, then you can understand better. One DNA molecule includes many genes. The nitrogenous bases in DNA pair up and form hydrogen bond. Adenine always with thymine, guanine always go with cytosine. Called complementary base pairing. We call that adenine always bind with thymine, cytosine always bind with guanine. We call that complementary base pairing. Complementary pairing can also occur between two RNA molecules or between parts of the same molecule. This you will see that later when you study transcription. In RNA, thymine is replaced by uracil, so adenine and uracil can form pair. This also I will cover up later. If I include too many detailed information in this macromolecule section, you might a little bit confused. Definitely this concept will come back later when we study DNA replication. So look at DNA double helix, two strands intertwined, and the string or strand is composed of sugar phosphate backbone. The previous slide said that, look at here, sugar and the fifth carbon of sugar molecule bind with the phosphate group. And this phosphate group will have a chemical bond of third carbon of sugar molecule of DNA. That forms sugar phosphate backbone. One end of DNA strand be end with here, 3' end of sugar molecule. The other end of DNA strand will have phosphate on 5 carbon molecule. So one strand of DNA has 3' end. The other end of that DNA strand has 5' end. That is one DNA strand, this DNA strand. The other DNA strand has upside down because nitrogenous base structure is ring, bulky structure. So in order to they form hydrogen bond between nitrogenous bases, the other DNA strand is kind of upside down. Therefore, the opposite end of 5' end of one DNA strand is what? 3' end. Counterpart of the end of this DNA strand, 5' end in counterpart position. So one DNA strand has 5' end here and the other end has a 3' end. The other DNA strand is opposite orientation. We call that anti-parallel. They are parallel, DNA strand parallel. Even though they intertwined, they are parallel, but orientation is one strand is opposite to the other strand. Therefore, we call that anti-parallel. DNA double helix, one end of this DNA strand has 3' end, then the other DNA strand has 5' end. The same thing. 5' end, the other DNA strand has a 3' end, we call that anti-parallel. And this string shows sugar phosphate backbone, and something protrude from sugar phosphate backbone is base. The base between two DNA strands form hydrogen bond that make DNA strand stay together in double helix. So you can see here, base pair joined by hydrogen bond. But transfer RNA has a single strand. RNA has a single strand, but sometimes a single strand RNA, they can fold to form double strand partially, not entirely. But DNA always form double helix entirely. We study 4 types of macromolecule, and this table summarizes the structure and function and what kind of a molecule belong to which category of a macromolecule. So you can screen and you can read. It's the same thing. Now, at the end of a macromolecule, I like to give a homework assignment. You can complete this homework assignment. So you go to grocery and investigate the nutrition fact on your favorite food. So when you buy one item, the outside item, always nutrition fact are written. And then you should write down that nutrition fact first on the paper, and then include your idea or comment in relation to carbohydrate, protein, and lipid to that item. But do not go by water. You write down on white paper, and it looks like, this is the example, some students like a Pop-Tart. Outside that item, report the nutrition fact first. You can do that. You just write down the nutrition fact written outside that item. And then you have to add your opinion. For example, 3 out of 4 macromolecules are present in the nutritional fact data: protein, carbohydrate, and lipid. In terms of lipid, both saturated and unsaturated fat are present. Saturated fat is the solid formation and unsaturated fat is the liquid formation. Although its obvious pop-tarts are not that great for you because saturated or unsaturated fat is not low to these students, but there's no trans fat and there's no cholesterol. Carbohydrate and lipid both provide energy. However, lipid store long-term energy. Lipids are necessary for the structural formation of a cell membrane, where a few carbohydrates occupy as well. So, in your comment, you need to write down some information related to 3 kinds of macromolecules, such as protein, lipid, and carbohydrate. Maybe some of your favorite stuff has more carbohydrate and very little fat, or some item might have a high protein concentration, and then your comment will be different. But you have to write down some information of protein, lipid, and carbohydrate in relation to your favorite item. You write down your name and your biology section number, and first you write down nutritional fact first, followed your comment. You will upload this on Canvas and then I will grade 20 points. This is all for macromolecule lecture.